All right, that looks like it's going. So I'd like to welcome everyone to today's edition of the Polyploidy Webinar. Today, due to a cancellation, we just have one speaker, but it should be fantastic. Up today is Alyssa Phillips. Alyssa comes to us from the University of California, Davis, where she is a PhD student, or PhD candidate, I should say. And she's a plant biologist interested in how polyploidy and genome size variation contribute to adaptation and population dynamics. As I mentioned, she's currently a PhD candidate at the University of California, Davis, and a U.S. Fish and Wildlife Service Directorate Fellow. Her dissertation research integrates population genetics and ecophysiology to study the demographic history of mixed-ploidy and big bluestem, a North American prairie grass. And today she's going to tell us about that work and this really exciting story on the origin and persistence of mixed-ploidy in Andropogon gerardi. With that, I'll let Alyssa take it away.
Thanks, Mike. Yeah, I'm really glad to be here. This is my first time presenting at the Polyploidy Webinar. So I'm very excited to get some feedback on this work. Yeah, so I guess I'll just jump into it. So I'm really interested in mixed-ploidy species, just generally. I think we often think about polyploidy as, a finished process, being fixed in a species. We think about it like the tips of the tree. But mixed-ploidy species are really common. There's about 16% of plant species are thought to be mixed-ploidy, and that's probably an underestimate. And a couple particularly well-studied ones are Arabidopsis aeruginosa and switchgrass. And then last month, we had a great talk from Ben Blonder on quaking aspen, diploids and triploids there, a lot of which resonated with my work, and I think is a great intro to what I'll be talking about. But the commonality of mixed-ploidy species is really surprising under the theory of minority cytotype exclusion. So this is the idea that you have, a population of diploids and a polyploid arises, and it's under a frequency-dependent mating disadvantage because if it cannot mate with the diploids around it, it doesn't have a mate, and it is under a fitness disadvantage, essentially. Despite this theory, there are ways in which real plant species and real mixed polyploid species persist. And this has been particularly well reviewed in these two recent papers, but I'll just highlight a couple here just to kind of like set the stage for what I'm going to talk about. The first being a mixed-ploidy species can overcome minority cytotype exclusion through recurrent polyploid formation. If you just have continuously produced polyploids, you have mates. Mixed mating systems are also useful. So if you can reproduce asexually, you have mates. Asexual reproduction can be useful. So for example, grasses reproduce vegetatively through a rhizome where they send out like identical clones through this underground stem. Interploidy reproduction, as well as ecological differentiation. So Justin Ramsey's work is probably my favorite example of this. And I see they're on the call here today. They were looking at tetraploids and hexaploids in wild yarrow, where the tetraploids live in this more mesic grassland coastal environment, and hexaploids live in this xeric dune environment next door. What they did was they transplanted tetraploids into this dune habitat and measured survivorship, and found that the hexaploids had higher survivorship in their dune habitat, suggesting local adaptation. But what they also found is when you take neo-hexaploids from the tetraploid populations and grow them in the dune habitat, that they have a higher survivorship than the tetraploid, suggesting that this first generation of polyploidy provides some kind of adaptive benefit to this new habitat. And so really, I'm just broadly interested in this question of how mixed-ploidy species persist, what strategies do they use, and how does that affect other population level processes? And I'm studying this in Andropogon gerardi. You might notice I'm spelling it with one I if you're familiar with this species. This is not an error. Some taxonomic nomenclature rules have changed, so it's switched from two I's to one I, just in case you're familiar with the species. This is a North American tallgrass prairie species, although it has a very wide distribution. It's found in also mixed grass and shortgrass prairie ecosystems. It's a bunch grass rather than a turf grass, so it grows in a clump rather than spreading across the ground like your turf grass in your lawn, for example. It's a long-lived perennial. species, and it commonly undergoes vegetative propagation through a rhizome that I described earlier. And it's also an obligate outcrosser, so it's actually selfing-compatible, so selfing isn't really an option here. It is also very ecologically important. So makes up 80% of the annual net primary productivity in tallgrass prairies, which is a very threatened ecosystem. This in the light green here is the historical range of tallgrass prairies in North America, and the green is the remnant prairies, only which 4% are thought to really remain, and they're highly fragmented. So trying to understand population level dynamics in big bluestem, as well as genetic diversity and the adaptive potential of the species can tell us a lot about the future of tallgrass prairies over the coming years under our continuously changing environment. As a polyploid species, my favorite trait of the species, there are two common ploidy levels, hexaploid and a neoploid. Chrissy McAllister found that both are found across the range, about 50% for each. So they're equally common, but they're not equally distributed. So populations in like the Southwest, the more arid climates have a higher concentration of the 9X cytotype than do the more mesic prairie ecosystems. But it's really unclear why the 9X are so common, and also what is explaining this distribution, especially because the 9X, or I should also, before I get into that, if you look within a population, they're where they're growing sympatrically, they're pretty equally, Kathleen Keeler found they're pretty equally dispersed within the population. So there's no like microgeographic niche separation. So it's one of the big questions is why is this happening, and why do we see this distribution, and why are they so common? This is especially surprising given the 9X individuals are sterile, or they're effectively sterile. So here we're looking at the counts of bivalence and multivalence in the hexaploids and the aneuploids. And what we see is that the 9X individuals have these common meiotic irregularities. They have all kinds of multivalence and univalence forming, while the hexaploids are, they've mostly form bivalence. And these common meiotic irregularities lead to the 9X individuals generally having less viable seed, as well as the seed that is viable are aneuploid that have a shorter lifespan. So they maybe only survive a couple years, like 3 years on average. And we also know that the self-incompatibility seen in the hexaploids is still maintained at the 9X level. So there really isn't a viable sexual reproduction mechanism, it seems, in the 9X individuals. So why are they more common in the arid environments? And why are they so common in the first place? There's a few hypotheses. We have a few hypotheses for why this might be, although I don't think this is, I think there could be more options than this. But it could be due to a difference in fitness, where the hexaploids could be just like more fit in the mesic environments, or the 9X are more fit in the arid environments. Alternatively, it could have something to do with the rate of whole genome duplication in different parts of the region. There could just be more polyploids formed in the Southwest. It's really unclear, but I think we need a lot more information about the system before we can dive in and test these specific hypotheses. And so the question that the first chapter of my dissertation is focused on, and what I'll be talking about today here, is how many times did the 9X cytotype originate? Where did it come from? To try and get at this question of, why is it so common? And I'm going to be answering this question using some pretty common population genetic metrics, and also a little bit of quantitative genetics. And really, to answer this question, there's two possible answers. One is that there's a single origin of the 9X cytotype. If we think about what genetic diversity would look like, then you would have genetic groups that are composed of a single cytotype. So on like an unrooted tree, we would have groups composed of one ploidy level. Alternatively, you could have multiple origins, and so we would expect to see genetic groups composed of multiple ploidy levels. And given the cytology and the life history of the species, we would expect that the 9X are probably have multiple origins, and that they're maintained by recurrent polyploid formation. Chrissy McAllister started answering this question with about 1,000 GPS markers and found that there's at least 3 origins of the 9X individual across this very widespread species. So she found there's 3 primary genetic groups, maybe 4, 3 primary genetic groups in which the 9X individuals cluster, but 1,000 GPS markers wasn't really high enough resolution to look at the population level or zoom in within that region. And so that's really what I'm interested in, is zooming in and saying what's happening at this population level. How many times did the 9X form within a region? And even within a population, how many times did they originate? And to do that, I'm using a data set of 27 populations from across the range, both a temperature and a precipitation range. That's low coverage whole genome sequence data. The white populations are, we only happen to sample hexaploids, and we have 3 populations with a good sample size of mixed-ploidy species, and those are marked in black here. We also have a beautiful new reference genome, thanks to JGI and Hudson Alpha. John Lovell's on the call here. That was a part of this team. We're super grateful to have this beautiful assembly. They were able to resolve all 3 subgenomes of the hexaploid and phase the subgenome. So we have 2 haplotypes here. It's a Centinely plot against sorghum. And it's been an incredibly useful tool. And we're taking we're using a new to us for sure genotyping approach to do this. And we're calling it kind of like a single read genotyping approach. And this is implemented in Angst. And I was just going to briefly talk through what this is. This method has typically been used in ancient DNA, where they have very low coverage genome sequence data, because oftentimes your low coverage genome data can be biased by coverage a lot. A difference between 1x and 5x coverage can really skew your genotypes. And so that's what this method was developed for, and we're kind of co-opting it for potentially using it for polyploidy. So imagine you have a chromosome and you have a reference SNP or you have a SNP where the reference allele is A, and you have 4 reads that are aligned to that position, 2 of which are A and 2 are B. If we called a normal genotype and a diploid, it'd probably be AB. And a tetraploid, it might be some kind of heterozygous genotype. We have pretty low coverage, so it'd be difficult to resolve. But what we can do is we can randomly draw a read from this site, and if it is the same as the reference allele, we can give it a 1. And if it's different, we can give it a 0, and we can genotype at every site and every individual. And if we can sum these genotypes over the number of sequence individuals to get our population allele frequencies. And this turns out to be pretty robust to ploidy differences. So we simulated 5 populations, 10 genotypes per population, 5 diploid, 5 triploid, with an FST of 0.1. And with regular genotypes, what you get when you run a principal component analysis is kind of like this artificial clustering of diploids and triploids. And although all these individuals were sampled from the same population allele frequency, this clustering happens because of excess heterozygosity in the polyploid genotypes. And this is a known common issue. With a single read genotyping, we don't get these artificial clusters. And that's because we're using like a continuous, a binary genotype value rather than polyploid genotypes, essentially. And we're being really cautious moving forward with this approach as it's new. So we're simulating data for each of the methods we use to try to understand where it might be biased and what the limitations are. But so far, it seems really robust in the last couple of months, and pretty excited about this going forward. And so this brings me back to the big question we're trying to ask: how many times did the 9X cytotype originate? And we're really going to answer this question primarily using population genetic approaches. The first question I wanted to ask is, what is the broad population structure of our sequenced samples? And so using our single read genotypes, I ran a principal component analysis. And we see that individuals pull apart into 3 broad groups. The colors here are the populations. There's too many populations to color anything in a useful way. But if we look on a map, what we see is that this cluster here is roughly all of these eastern populations, while we have this like western group of populations, and then these 3 populations that pull out kind of separately. Our 9X individuals are triangles here, and you can see they all fall into the same region. And so that's the region we're going to be focusing on to answer the question of how many times did the 9X population originate. It's just looking within this western region. Briefly, I wanted to highlight this like odd group here and what's happening. If we run structure here, I'm just showing a subset of the populations. K=2 is best supported, and we get our west and our east groups resolving pretty well. And New Mexico, which is our representative in this odd group, is falling into this western cluster. And I can talk more about that later if folks are interested. But we do broadly have these 2 big regions, which is similar to what Christy McAllister was finding. But so we don't really know what's happening within here, and the PCA doesn't really have, and structure doesn't really quite have the resolution to tell us about this population differentiation. And so to look at differences between populations, I'm going to I estimated pairwise FST between populations. So briefly, for folks that aren't familiar with FST, it's looking at the proportion of heterozygosity that's shared between 2 groups. So if you have a higher FST, more heterozygosity is within population, so is not shared by individuals, versus a lower FST suggests more shared heterozygosity and more relatedness between the 2 groups. So a lower FST is more closely related. And so under our 2 hypotheses, this is what we would expect. Under a single origin, we would expect that 9X individuals from different populations in this western region would have lower FST, be more related. Whereas under multiple origins, you would expect that the populations would be structured like the 6X and the 9X from Kanza would have lower FST and would be more related to one another. And so what we see is, I realize this slide's a lot, so I'm going to point out broad patterns to you. These are our Western populations. What we see is that just generally the populations are pretty homogeneous. I realize it looks like there's some color variation here, but our range in FST is actually very low. The average is about 0.1 is the differentiation between all populations, suggesting they're generally homogeneous. And here are the hexaploids, and here are the 9X individuals, and they're pretty equally differentiated from one another. They're not particularly related, suggesting that there are multiple origins of the 9X, that the Kanza 9X are not particularly related to the Austin, Texas 9X individuals. But the homogeneity of these populations and this differentiation really led me to ask what's happening within these populations if we compare individual genotypes to one another. What is happening? Are the 9X within a population related? And we can answer that using, we can ask this by estimating kinship between 2 individuals. So this is the probability that if you randomly sample an allele from one individual and then randomly sample it from another, that they are identical by descent. They come from the same recent common ancestor. So a parent and a child would have a kinship of a 4th, first cousins, a 16th, and with yourself it'd be 1/2. So higher kinship is higher relatedness. Under a single origin, we would expect kind of this pattern where more red is more related, where you would have the 9X individuals within Kanza would have higher kinship. And so we would have kind of this like within population structure occurring. Whereas in multiple origins, you would kind of have groups where the 9X and the 6X are intermixed with one another. And what we see is generally very limited population structure within the Western region. So these are our genotypes just in the West. And I have the 9X individuals marked in black, and the closer to red is more related. Very low relatedness amongst individuals. It's very homogeneous, which is also what we saw reflected in FST. This is our odd 3 populations in the West from the PCA earlier. They seem to just have some odd inbreeding occurring. But other than that, there's very limited population structure within the West. The 9X are intermixed with the 6X. So we don't see any particular clustering of the Kanza 9X with the other Kanza 9X, or even the Kanza 9X with the Boulder 9X. It's very intermixed with one another, with the exception really being this Austin, Texas populations. We don't have great sampling near this group. And so it's unclear if there's just higher relatedness in that region generally, or if these could just be more related to one another. But overall, they still have quite low kinship as to what you would expect from like siblings, for example. And so this kind of led me to think that the 9X are actually like neopolyploids, that they're likely their own individual, their own polyploid event. And I think the cytological data and the life history supports this idea as well. That this is what we would expect neopolyploids to look like. And so that's my broad takeaway from this talk, is that I think the 9X genotypes are likely neopolyploids, which suggests they would be maintained through recurrent whole genome duplication or clonal reproduction, and that the commonality of 9X individuals is likely due to this recurrent formation, as well as just the long-lived nature of the species. We really don't know how long a single individual can survive. But this opens up still more questions. So still, why are they so common? Can they just survive vegetatively potentially longer than 6X individuals? What is happening there? And what does this mean for the future of North American tallgrass prairies? If you have populations dominated by a cytotype that's effectively sterile, this is something we maybe need to be considering when we're thinking about restoration of this ecosystem. And I'm actually, the other part of my dissertation is a common garden experiment where I'm looking at ecophysiological differences between the ploidy levels to try to understand, is there some kind of difference in relative growth between species, between the cytotypes or between populations that could be explaining this pattern as well? And with that, I'd love to thank all of my collaborators. This has been a really big effort. Big thank you again to JGI and Hudson Alpha for assembling these genomes, or the genome, and my collaborators in Toby Kellogg's lab who collected the Andropogon samples. Could not have done it without all of these folks. And with that, I'm happy to take questions.
All right, well, thank you so much, Alyssa, for a really wonderful talk. I'll give everybody a minute or two here to pop their questions, either to raise their hand or drop a question in the chat. I'm gonna go ahead and ask a question while we wait. So I haven't thought much about how the species, is dispersed, and is it animal dispersed? Is it wind dispersed? I'm just thinking about, the structure of these populations and how things get around. What's the dispersal mechanism?
Yeah, I think that is a great question. I mean, I think they're wind pollinated, but I think some of the seed dispersal, some of the seeds can be carried a short distance through the wind, but I honestly haven't thought much about that as well. One of our concerns definitely was sampling from remnant prairies that weren't reseeded by by other restored prairies. And so I'm kind of wondering if that is an issue with one of these populations. One of them is in like Nebraska, and is very far distance. So we're concerned that maybe it was reseeded or there was introduced seed. But yeah, I haven't thought much about, I guess, seed dispersal side of things.
Yeah, it's very cool. I really like this though. It's really interesting to see this lack of structure across such a large geographic scale as well. I feel like this is similar though to like some of the other things, like some of Lauren's plots for like this Rhelianthus annuus that have been generated over time for the prairies too. It's a very different beast, very different species and species biology. But just thinking about the other things in that part of the planet, that those huge distances, they have pretty low FSTs as well, if I remember correctly. So, just sort of curious if that's something that we see generally in these really wide-ranging species like this, and then maybe kind of having to scale what we think of as a really low or high FST, right? For
right, like point one in some systems is quite. I mean, that's not a low FST.
Yeah, exactly.
What's surprising is that every pair that we decided to pair against one another had an FST of 0.1. I think that's particularly what makes me describe it as homogeneous.
Yeah.
And the kinship.
Yeah. Very cool. I see Justin Ramsey is his hand raised. Go ahead and just popped on.
I was great talk, by the way. That was great. I was curious, are the 6X plants multisomic, have multisomic inheritance? Is it fully multisomic? And is that a factor maybe in the low differentiation? Just that there's a lot of chromosomes interacting in meiosis there.
Yeah, that's a great question. So the cytology work I cited, cytological work, there are generally, it behaves with bivalent pairing. There are sometimes multivalents at a low rate. So it's bivalent enough that I feel pretty good saying it's disomic and treating it that way. But I am starting a side project trying to identify regions of the genome where there might be multisomic inheritance.
So even if it's bivalent, I mean, if there are any cytogeneticists here, feel free to chime in. But the question is if they're random, because so it still could be segregating in a hexasomy, but it's even more if it was multisomic. Or sorry, if there were multivalents forming.
Right. So that's a great question. So from what we understand about the subgenomes, it's an allopolyploid. So there are two subgenomes that are supposedly more closely related to one than the third. But the two have, it's thought that they were a tetraploid long enough where they differentiated before hybridizing with the diploid. And that's also what this project I'm starting to look at is going to explore a little bit more to see if that holds up. But so far, the work coming out of Toby Kellogg's lab has supported this idea that the three subgenomes are quite differentiated. And I think through our discussions with John, and also there is a second genome assembly as well, those collaborators, the way the genome assembly comes together really supports the idea that the three subgenomes are distinct because you can assemble them separately. So
interesting.
So I'm feeling okay about the three distinct subgenomes at the moment, but we'll see. Yeah, if it helps.
Oh, no, that's great. It's an interesting system, and I'm sure the genetics are very complicated. So trying to work with that while trying to use the molecular data is a challenge. So
yeah, absolutely. It's really cool. So here we'll go through, we've got a little pile of questions. We'll let George Tiley go first, and then we'll go back to the chat to Rob's question next. So George, go ahead, and then Rob.
Hey, yeah, that was a great talk. Thanks for that. Two questions. One, we see, as you point out, like a lack of structure, and it looks kind of one continuous gradient of variation. And you have these nice pairwise FST results. So have you looked at isolation by distance? Is just pure physical distance the primary explanation of the FSTs?
Yes, that is next. That is what I am exploring now. So these results are relatively recent in the last couple of weeks. Just trying to, I've been spending a little bit of time trying to think about what this means and what kind of questions are useful next going forward. If they are neopolyploids, what is now interesting to think about in terms of adaptation in the species. And that's the first thing I want to look at. So right. I think that it may or I don't know. I'm not sure if that's going to be it. Just because, yeah, I'm not sure.
Yeah, but it is like kind of a thing to do.
Mm-hmm.
To see what happens. And then a question on the actual genotyping process. So you mentioned you use angst to essentially reduce the complexity of these allele frequencies to zeros and ones. And I'll just say I like genotype likelihood methods in general. It lets us impute missing data and deal with a lot of uncertainty. But was that choice just because you don't trust our ability to get allelic dosage right and call genotypes at like 6 and 9x? Is it just kind of throwing darts at a dartboard at that point?
Yeah. This is what I've definitely spent most, if you're breaking down the percent of time I spent on this project, most has been genotyping and calling SNPs. So what's nice about our reference genome, just to start, is that because we have all these subgenomes resolved, we can just genotype as diploid and triploid genotype. So if we were going to call genotypes, that makes it a lot simpler. But I've played around with, I spent a lot of time working with genotype likelihoods. And when I finally got to running a PCA, PC1 was a giant smear completely explained by coverage, because our coverage, although it's like low coverage, the range was from 0.5 to 5x coverage. And that is, that's a huge difference in coverage, 5-fold. And so all of the confidence in your genotypes then is explained by coverage. And so that's when we kind of turned to this approach, which was specifically designed to deal with that coverage issue. And it seems to be reliable for polyploidy as well thus far. And I think that's kind of how we got there. I do trust genotype likelihoods, but if you have a big variation in coverage, it doesn't work. And there wasn't a way to subset our samples. We would have had to throw out most of our samples or subsample 5x coverage to 1x coverage to make genotype likelihoods work. And so this actually kept more information using this approach.
Yeah, thank you.
Rob's question here in the chat is, did Kathy Keeler investigate the lifespans of the 6x and 9x?
Yeah, so I don't think like lifespan, but she has measured relative growth. Someone can correct me if I'm wrong about the lifespan, but I know she did look at relative growth, and that the 9x did have higher relative growth, like year to year. I am also seeing that in my Common Garden data. Spoiler, very exciting, very preliminary, but between just 2 years, right? Just sampling year 2 versus year 1, the hexaploids doubled in size, while the 9x individuals seem to have tripled their size. And so we also have measures of like number of tillers, biomass. That's just looking at biomass, and then also like circumference of the plant to see if they're expanding outwards. And so we haven't explored those yet, but just in like sheer biomass. But visually, you cannot tell a difference between the two ploidy levels. They look identical. So that's why you're sampling. It's hard to sample evenly.
Well, I'll just answer for Rob. That's really exciting. Anyway, Move on to the next question here from Pat Heslop-Harrison here in the chat. Pat, if you want to hop on, feel free to do that. You got a long one.
Yeah, sure. Just a couple of questions about whether there was structural variation that you detected between the different sets of chromosomes in the polyploids with the sequencing. And it's great that you could tell the homeologs apart so well.
Yeah, I see, John, you replied in the chat. Do you want to answer that?
Yeah.
Everyone? You definitely are the better answer to that. Yeah.
Yeah, sure. I think you pretty much nailed it. There's one subgenome that really easily fell out with, we do like a shared Kamer approach to phase subgenomes.
Yeah.
First of all, these two haplotypes split extremely cleanly, which means that it's a very heterozygous individual. And then also in the homologous chromosomes, we always got within each haplotype clusters of 3. So we never saw anywhere where there was a cluster of 1 or 2. That's what you would expect if there was homogenization due to non-homologous recombination.
Did you see any interchromosomal translocations between those clusters, between those members of each of the clusters of 3?
Right. So that would be indicative as well of exchange, even if you still separate them.
And that would complement what the question is about the meiosis, what it would be as well.
Yes, we don't see any evidence of that. There's no position, but at the same time, 2 of the subgenomes are difficult to distinguish. When we did the initial clustering, we got 10 in one cluster and 2 in another. And then we had to do a second clustering within the second pair to distinguish the other 2. Again, consistent with Alyssa's assertion that it's a auto-allo or has structure in terms of the diversity of the ancestors.
So all your survey sequencing was what about 10 gigabases of Illumina? For the genotyping. Sorry, did I miss it?
Sorry, could you ask that again?
Yeah, okay. So this is based on Hi-C and nanopore, or as well as Illumina, or what?
On the genome assemblies?
Yeah.
These are Hi-C integrated hi-fi assemblies with like 95x per haplotype hi-fi. It's a whole new world now, especially now that we get Revio. We have incredible coverage of hi-fi on these, and also I think this has like 180x Hi-C integrated. So we're sure that each chromosome, and I can pull it out. I don't have the stats in front of me.
No, that's great. And for the 27 lines that you had the survey sequence for, that was. How much survey sequence did you use? And well, I suppose the key question is this would you now use survey sequence rather than any type of GBS or?
Yeah, that's a great question. So the whole genome sequence data.
This one.
My analyses. So I end up with like 11 million SNPs. That are with very strict filtering, or what I think is strict filtering. And then to run most of these analyses, I subsample it down to 100,000 random SNPs.
Okay.
So, like you can do so much with 50,000, 100,000 SNPs. That's still way more than you're getting from GBS data sets. And the cost of low coverage whole genome sequence data is comparable to is comparable to GBS data. And so I think there's a real argument about switching to low coverage whole genome sequence data from GPS. I think that's how things are moving.
Yeah, absolutely. And also the sheer amount of time of preparing the samples and so on is 0 for the whole genome and substantial for any other selection.
Yeah. I think the challenge is going to be the accessibility of analyzing low coverage whole genome sequence data right now.
So you've got about 10 gigabases of sequence for each genotype, or how much?
There's about, I mean, it's like 1 to 5x coverage. I don't know. Yeah.
Okay.
Yeah.
Great. Okay. Thanks a lot. Great seminar. Thanks, John and Alyssa.
Thank you.
All right, yeah, there's a question from Allison. Does the assembly have centromeres? And if so, how do they vary across the subgenomes? And I don't know if Allison, if you want to jump on or not.
I don't
Give me
need
a
to say
second, I'll
anything
get the
else, but
stats
because
for you.
John's talking all about the assembly, then I'm like, well, so, but does it have centromeres? And because of course that's interesting for the subgenome dynamics. So I'm just curious.
Yeah, I'll pull those stats down. I'm just getting them off the server right now. I can
But
say
so they're
for
there,
sure
I guess. If they're
that
stats, there's a centromere.
the vast majority of those chromosomes are sequenced straight through the centromere.
Yeah.
And I'll get you the number that are fully telomere to telomere in a single contig in a second. But it's very high. This genome's great.
Yeah.
It's really, really clean. I bet we have full centromeres for 20 or more of these chromosomes with a gap in a few of them.
20 or more. Okay. And I'm looking at the figure. So it's 10 chromosomes, 3 subgenomes, 30 total.
So
okay. Okay. Yeah. Gotcha. Yeah. 30. Yeah. Okay. Cool. Yeah. No, I mean, it's, I think that would just, since you have such a nice assembly, that could also be a cool component to look at, like in addition to, some of these questions about homologous pairing or not. But if you've got centromeres across the subgenomes for any positions, then that would be interesting to see. I mean, I don't know either. And I know in a lot of these cases, we don't know what the centromeric repeat would normally be anyway. So A is defining it, but then if there is variation, which you might catch some of it, that would be interesting to see what those patterns look like across the subgenomes. Yeah.
It's a great suggestion. Yeah, I think we're definitely, I am transitioning more to thinking more about what questions do we want to ask at, the species level polyploidy versus within species polyploidy.
Right.
And we do have a second genome assembly. It's not quite of this quality. It's not chromosome scale, but it's very nice. And combined with this phenomenal resource. So hoping to potentially use these 2 assemblies to date divergence of the subgenomes.
Yeah, that's really cool.
And we have genome assemblies for really relatives to the ancestors of these subgenomes, but they are also have their own complicated polyploidy going on. So unclear how useful they will actually be.
I see. Yeah. Is you said that the other assembly, it's of another hexaploid or?
Yeah, and from a different region. Okay. So this is from the center of the range in Kansas, whereas the other assembly is from North Carolina.
Okay, cool.
So that's fun too. And that one, I should say, the big difference between the quality of the 2 assemblies is the high C data.
Sure.
The other one was not, we didn't sequence with high C. It was just hi-fi and bio-nano.
Oh, I see. Okay. So this one has high C, but the other doesn't.
Yes. Yeah. And so that really made all of the difference when dealing with the heterozygosity of the species.
Totally.
Yeah.
Very cool.
So I have that number for you. We have 10 of the 30 in HAP1 and 8 of the 30 in HAP2 are single contig telomere to telomere.
Very cool.
So that's a full, complete representation of a single biological haplotype.
Sure.
The others, most are 2. There are a few that have 3. Our least contiguous chromosome has 6 contigs.
Sure.
So in general, you're going to get most of the centromere, but maybe not all of it.
Yeah. But at least in some cases, there would be overlap where you could look at the different subgenomes and compare the centromeres there.
Yes, absolutely.
Cool. Thank you.
All right. Oh, I see Justin's got his hand up. I was going to ask a couple questions at the end there, but I'll let Justin go first.
And this may be wrong. I kind of remember there are other Andropogon species, maybe more in the south and the east of the US. I mean, is there any indication that big bluestem kind of exploded into its current distribution, that that could be contributing, sort of rapid expansion colonization could be contributing to the low structure?
Yeah, it's a great question. So crediting John again. John, I'm so glad you're here today. I talked to John like, oh God, he was visiting Davis a couple weeks ago, and we just happened to sit down and I showed him these results and he was like, do you think that the 9X are remnant from like colonization of the prairies after the retreat of the glaciers? Like, that would make sense. That's why they're left in the Southwest, potentially. If you can reproduce through vegetative propagation, that's great, but it's still not going to get you as far as if you can reproduce through vegetative propagation and spread your seed. So you won't be able to colonize as quickly. And still need to think about how we can ask questions about that, which is why I didn't talk about it today, because still need to think about how we would test that. I think we can, especially given, at least in the hexaploids, potentially looking at like range edge stuff, because we have the sample size for that, but really need to think about how there is a subspecies. Okay, our subspecies may be separate species, Andropogon hallei, it's like a sand dune big bluestem or sand big bluestem. I think it's unclear if that's a separate species. But I don't know if we have any samples of that we sequenced. I would have to check with Toby about that. But that's what I know about big bluestem in the region. Yeah.
Mm-hmm. Yeah. So that's really interesting. If you ever need stuff from the more northwestern edge of the range, we'd be happy, because we have hallei here in sand dunes in South Dakota. Okay. And also, even though we mostly are mixed and even shortgrass prairie, if you go to a wet enough environment, it's definitely here up in North and South Dakota, as I'm sure you know. So if we could ever help, let us know. We'd be happy to.
Yeah, that's great. I think there's just so many fun questions to ask about this system. Yeah, that's really great to know. Thank you. Yeah.
All right, looks like John has his hand up, and I'm going to ask a question before you go. But
sweet. I'll keep it quick. The parallels between your talk today, Alyssa, and Ben's from a few weeks ago are obvious and really strong. And, Ben was harping on the idea that, triploids probably aren't doing a ton of sexual reproduction, but these plants live for a really long time. There are lots of them, and who knows, they might get lucky. And so when I was thinking about your, sort of straw man hypothesis, single versus multiple origins, the second that you show that there's anything other than a single clone across all of them, you now know there's multiple origins. But then the question are these actually somewhat distinct evolutionary lineages? And my thought was, okay, well, that's really easy to answer, because it's just going to be a fixed set of haplotypes, and you're not going to see any recombination at all, or any signature of variation within a group. It should be all identical. But I mean, it's possible that, if these things live for 100 years and they try and do meiosis, several million times, one time it might work, and they could make a seed that's just fine, even if all of the evidence shows that it's not likely on a human or a single experiment timescale. That's all.
Yeah, I completely agree. Right. And, the only reason I have kind of separated out that I am kind of eliminating this gene flow hypothesis is because of the cytological work, and because time and time again we see the aneuploids. They did a lot of crosses between ploidies within ploidies, and it's just consistently aneuploids, and there's always a chance, right? And it's very, very small, but over infinite generations that's possible. And this is something I've talked with my PI a lot about. It's hard to eliminate that, right? So in the end, the kinship matrix could be neopolyploids, but it could also be just continuous gene flow between ploidies. And I don't know if we using these approaches can actually answer. We would have to, it would be really great to have a known hybrid you could sequence and then compare what hybrids look like. Now I'm thinking about it, that'd be really cool. But yeah, so I think it's a possibility, but I am putting it in a corner there and saying it's incredibly unlikely. And that could be a fault on me. Yeah.
Very, very cool. I'm going to jump in here and to wrap up. But as you were talking, I also was thinking about the ages of these different ploidy levels and things like this. Have you thought about doing something with demographic analyses or something like this to do modeling? I know that we've thought about this in in polyploids.
Okay, wait. Now, Alyssa, you're going to have to tell Mike about your adventures in demographic.
Oh gosh, that's a whole other thing.
Oh, okay.
Yeah, so the second we got the email about the genome assembly, we were like, sweet, where can we download it? And then Jeff Rosabar is my PI. He was like, can you align the genome to our other genome, all the haplotypes versus all haplotypes, so I can run FastSimCol on the subgenomes to look at divergence between them. And the very rough estimate is you have one genome that diverged 20,000 years ago or 20,000 generations ago from the other two, and the other two diverged yesterday in comparison. But that's like a very rough.
Yeah, that makes sense.
First look. And I think it's really cool, and I want to explore that more. But Jeff was like, send me these now. I'm gonna press go.
That's very cool. I mean, you see that a lot though, right? I mean, especially with this kind of, and I don't know, what all the sort of substitution rates and things are like, but where you get those, you can imagine you've got one divergence that's really old and then something more recent for the autos or something, I guess, depending on, the order of events. Yeah, we thought about this. Paul Bleshak has a version of Dottie for doing this in polyploids and accounting for inbreeding and stuff that might also be really fun to check out. I would be curious how well what it would say.
Yeah, I would be interested, just particularly running it on the two genomes that are more closely related.
Yeah.
I think we have to treat those separate to get any one of
the nice things that Paul did is he implemented some models in there to distinguish regions, to sort of tease apart allo and auto, right? To try to let the data tell you how it's behaving in terms of the modeling. And so that might be useful here too, where if you're not really sure which parts are, you don't have to do much work yourself to figure out and classify those subgenomes then. Let the allele. I
see that the Adam's in the chat. Adam Healy is running the EDTA to get a TE annotation for me because I was inspired by Adam Sessions' talk 2 months ago. So I could look at TEs that are different between subgenomes and also use that. So that's like my fun side project right now, where I'm trying to look at if there's like unique TEs to the subgenomes, and we can also potentially get an age estimate from that approach as well, which would be cool.
Yeah, it's very cool. This is really cool work. Well, I think that we've definitely ran over. Well, we haven't really run over our time for the meeting, but I think it worked out pretty well that there was a cancellation today, and lots of questions, and this is really wonderful to see all this really great work. And I thank everyone for sticking around and asking questions, and thank you, Alyssa, for tolerating it. And hopefully we'll see you. I'm supposed to announce as well the Polyploid conference again. So hopefully we'll see all of you or a bunch of you in Florida this May in a couple of months. Pam asked me to just sort of point out that it's now open for submission, and that the registrations are not expensive. And I'll say as well, I think they're like $100 for students and postdocs. And the meeting accommodations are also very, very, very reasonable. So for anybody that wants to come and hear about polyploidy in person, that meeting is May 9th to the 12th in Palm Coast, Florida. So, all right, yes, and Allison is saying that we also submit abstracts to SMBE. I know there's going to be a polyploids symposium at BSA this summer as well. What else is happening? And we've got we have the polyploidy special issues of apps and AJB, which I know some of you on the call, many of you have submitted proposals for. We're going to get around to reading those soon. They've been slowly trickling in here over the course of the last few weeks, and we're just now getting ready to get organized to send out those, to pick the papers and send out notification. But otherwise, we'll have one more Polyploidy webinar here for this academic year, and then on May we'll all be in person. But our next two talks will be Sritama Vadra and Navita Padilla Garcia, telling us about polyploidy in their work and a variety of systems. And I hope everybody can show up again. That'll be on April 5th, the first Wednesday there in April, again at 11 a.m. PST. But again, I want to thank you, Alyssa, and everyone else for sticking around and asking great questions and really, really cool, thought-provoking work. Thank you.